Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing next exam biology section question paper for the year 2023 shift 1. If you are new to my channel, please do like, share and subscribe. Coming to the first question. A cladogram representing the evolution of some animals P, Q, R, S is shown. 1 to 4 is in the cladogram represent different features that have evolved. The correct statement with respect to the given cladogram is. So if Q is a bullfrog, 2 could be placenta. So this is Q. So they say that Q is a bullfrog, then 2 placenta. So placenta evolved in bullfrog. Bullfrog doesn't have placenta. So that is not correct. If features 3 and 4 are mammary glands and hair respectively, then R represent kangaroo and 5 and S represent humans. So 3 and 4, these two, they represent, 3 represent mammary gland and 4 represent hair. Then R should be kangaroo and S should be human. R representing kangaroo because it is a mammal and mammary glands have evolved in kangaroo. So that feature is correct. But hair is the feature that led to the evolution of humans is not correct. So second option is also wrong. If Q, R and S represent bullfrog, koala and human respectively, the feature 2 could be pair of limbs. Yes, pair of limbs evolved in bullfrog. Then it can be seen in koala also as well as in human. So that option is correct. If feature 1 is lungs, then P and Q are shark and bullfrog respectively. P cannot be shark because shark do not have lungs. So the correct answer is option 3. Then next one. A 1000 base pair double-stranded DNA B form has a melting temperature of 58 degrees. If a duplex RNA, that is A form of the same length and sequence is constructed, then the TM of this new RNA duplex with respect to the DNA would be. So the answer here is it would be higher due to greater stability of A form of RNA duplex. So answer is first one itself. Then next one. A biochemist want to purify protein X molecular weight 30.2 kilodalton and pI 7.5 from a solution containing proteins X and Y. Y molecular weight is 30.9 kDa and pI is 3.5 using ion exchange chromatography. In this technique, an anion exchange resin is positively charged and a cation exchange resin is negatively charged. The most appropriate resin where protein X but not Y will remain bound is. So before coming to the answer, I would like to tell that the isoelectric point of a protein, it is defined as the pH at which net charge on the protein molecule is 0. And accordingly, proteins are positively charged at a pH below their pI. That is, if pH is less than pI, then they are positively charged. And because of their positive charge, they will bind to cation exchange resin. And negatively charged at a pH above their pI. So if the pH is above their pH is greater than pI, then the protein will be negatively charged and it will bind to the anion exchange resin. Now they want to know where for protein X to bind, not protein Y. So cation exchanger at pH 7.5. So protein X is having a pI of 7.5. So at pH 7.5, the pH will be equal to pI and no net charge will be there. So it will not bind at all. So option 1 is not correct. Anion exchanger at pH 2.5. So here pH is less than pI. pH is less than pI. I told you it will be positively charged. The protein will be positively charged and it will bind to a cation exchanger and not an anion exchanger. So option 2 is also not correct. Then pH is equal to 5. Now here pH 5 is less than pI, that is 7.5. So it will be positively charged and it will bind to cation exchanger. So third option is correct. Similarly, here also just like the first option, anion exchanger at pH 7.5, Pi is equal to pH, so no net charge and it will not bind. Then next one. Bacterial endospores are highly resistant to environmental stressors such as heat, UV radiation and oxidizing agents. They can remain dormant for a prolonged period. During the period of dormancy, they prevent the accumulation of potentially harmful mutations in their DNA. This is primarily due to, decreased water content and reduced enzymatic activity in the bacterial endospores. So answer is option 1. Then next one. Pea seeds were allowed to germinate for 4 days and segments of the epicotyle around 1 cm were cut and placed in a nutrient solution having pH 7. These epicotyles were then treated with cyanide at a concentration of 0.1 millimolar and the membrane potential was recorded. Cyanide was washed out and the membrane potential was further recorded up to 78 minutes. A graph of cell membrane potential against time was plotted as shown. So at this point over here, 0.1 millimolar cyanide was added, then respiration decreased or cell membrane potential decreased, then when cyanide was removed it started to increase. So based on these experimental observations, the correct option is, so what happens after cyanide poisoning? So after cyanide poisoning, the electron transport chain will not be able to pump electrons into the intermembrane space. So the pH of the intermembrane space, it would increase and the pH gradient would decrease and ATP synthesis would stop. Now why does ATP synthesis stop? Because without a proton gradient, protons will not flow through ATP synthase, hence no ATP will be produced. Now first option, addition of cyanide will decrease the membrane potential because of the depletion of ATP. I already told that. So first option is correct. Addition of cyanide will decrease the pH of the intermembrane space. It will not decrease, it will increase the pH of the intermembrane space. That also I told just now. Addition of excess oxygen will increase the membrane potential in the presence of cyanide. That is also not correct. Addition of cyanide will cause a rapid but irreversible depolarization of the membrane potential. That is also not correct. So the correct option is option 1 only. Then next one. In the given pedigree, circles represent females, squares represent males. Filled shapes indicate affected individuals, while unfilled spaces indicate unaffected individual. Based on the pedigree information provided, the correct inheritance pattern is. So here you can see, if there is no, if the parents are not affected, the children could be affected. Here one parent is affected, but all the children are not affected. Only few of them are affected. Here both the parents are affected and all the children are affected. So, and there is also what is called as skipping of generation. This trait, it doesn't appear in generation 2, but the kids born to the parents of generation 2 are affected. So there is skipping of generation. Then both males and females are affected. So there is no, so you can happily rule out X-linked dominant and recessive character. Even if parents are not affected, children could be affected. That I already told. So the option 2, that is autosomal recessive condition, is the inheritance patterns followed in the pedigree. Then next one. A population has a single locus with 3 alleles X1, X2, X3. The frequency of these alleles are P, Q, R respectively, and if P plus Q plus R is equal to 1, then the correct statement is, the population is in Hardy-Weinberg equilibrium. The population of the allele must meet 5 rules in order to be considered under Hardy-Weinberg equilibrium. That is, no gene mutation, no migration, random mating, no genetic drift, and no natural selection. But these conditions are not mentioned in the question. So the information is insufficient to state whether the population is in Hardy-Weinberg equilibrium. So your answer will be option 3. Then next one. Mendelian theory of inheritance was crucial for Darwin-Wallace theory of natural selection because it explained how new variation will persist in a population over time despite their low frequency, which is essential for natural selection. So the answer here is option 1 itself. So coming to the next question. In closed ecosystem, red algae, crabs, green algae are present and interact with each other. In order to understand the interaction between them, the following experimental treatments were imposed. All crabs removed, all green algae removed, all crabs and green algae removed, all crabs, red and green algae are retained. The result of red algae growth from these experiments are shown in the graph. Plus indicate presence and minus indicates absence in this closed ecosystem. Red algal growth in all the treatment was significantly different for each other. So this is with crabs, this is without crabs, this represent green algae present, this is green algae absent. Similarly here also green algae present, green algae absent, and this is the growth for red algae. Now coming to the options. In the absence of crabs, that is over this graph, green algae positively affected red algal growth. Green algae negatively affected. If you see this bar diagram, it shows that in the presence of green algae, red algal growth is very less, whereas in the absence of green algae, red algal growth is more. So this is incorrect. Then next one. In the presence of crabs, that is over here, this graph, green algae positively affected red algal growth. So green algae present in the presence of crabs, green algae positively affected. No, green algae negatively affected because in the absence of green algae, red algal growth is more when compared to presence of green algae and presence of crab. So it negatively affected. So that is also incorrect. Then next one. In the absence of green algae, that is you have to see for the black bars, crabs negatively affected red algal growth. In the absence of green algae, crab negatively affected red algal growth. So in the absence of green algae and in the presence of crabs, that is this graph, this bar diagram, crabs negatively affected red algal growth. So if crabs are present, red algal growth is lower when compared to green algae absent and crabs absent. So in the absence of green algae, crabs negatively affected red algal growth. Why? Because if you compare these two bar diagrams, you will see that this is in the presence of crabs and absence of green algae. This is absence of crab and absence of green algae. So they have told for in the absence of green algae and crabs present. So you have to see for this bar, compare it with this bar. So crabs negatively affected red algal growth. That is correct. So if crabs are absent, it will positively affect red algal growth. Then next one. In the presence of green algae, now you have to see for the white one. Crabs negatively affected red algal growth. No, crabs positively affected red algal growth. If you compare these two bar diagrams, you will see that in the presence of crabs, it positively affected red algal growth. So the option 3 is correct. Then next one. The permeability of molecules across a lipid bilayer depend on their size, solubility in the non-polar solvent relative to their solubility in water. For the species oxygen, water, K plus glycerol and glucose, the order of their permeability across the lipid bilayer in the absence of any protein transporters is. So you know that the semi-permeable membrane, gases will diffuse readily. So oxygen will diffuse faster, followed by water, then glycerol, glucose, and then lastly ions. Here the ion is K positive. So option 1 is correct. Then next one. A monoclonal antibody specific for a given epitope of an antigen. IgG antibody, it is labeled as M, was produced in the laboratory that bind to the surface protein P of a virus V1 and neutralizes 100% of V1. So this IgG antibody, it will bind to the P protein in a virus and virus V1, and it will neutralize the virus V1 100%. Then it was observed that M is also effective in neutralizing other viruses V2, V3, V4 at 40%, 90%, 32% efficiency as compared to V1. If there are no confounding factors, then the most accurate inference is. So first option, V1, V2, V3, V4 have identical surface proteins. If it was identical, then M would have neutralized it 100%. But only V1 is getting neutralized 100%, whereas V2, V3, V4 are 40, 90 and 32 respectively. So first option is not correct. Then next one. The genome of V1 and V3 has to be 90% identical for the observed binding efficiency. It is not the genome but sequence of P has to be identical for the observed binding efficiency. Then next one. The sequence of P is more similar in V2 and V4 than in V1 and V2. The sequence of P is more similar in V2 and V4 as well as in V1 and V3, not V2. Why? Because V2 and V4, they are almost somewhat similar in neutralizing effect, that is 40% and 32% respectively, whereas V1 and V3 it is 100% and 90%. So you can say that the sequence of P is more similar in V2 and V4 than in V1 and as well as in V1 and V3, not than in V1 and V2. Then last option. A segment of sequence P is highly conserved in V1 and V3, whereas the same segment is less conserved in V2 and V4. So yes, because that is why V1 and V3 are getting neutralized at 100% and 90% neutralization activity is shown. Whereas V2 and V4 only 40% and 32% efficiency is shown. So segment P is highly conserved in V1 and V3, whereas the same segment is less conserved in V2 and V4. So the answer here is option 4. Then next one. A genetic engineering experiment resulted in a new hemoglobin variant which exists primarily as alpha-beta dimers in solution. Based on the oxygen saturation curves of the variant and normal hemoglobin, it can be inferred that. So what you can see from the graph, the variant displays a regular curve. As you increase the concentration of oxygen, variant becomes saturated very quickly. See, this fast it is getting saturated with oxygen and then levels off. See over here nearing to 100. Whereas this implies that variant has high affinity for oxygen. Whereas hemoglobin, it displays a sigmoidal curve like this, which means it has lower affinity for oxygen and binds oxygen relatively weakly. This type of curve, it is the result of cooperative behavior of hemoglobin. So we will see the options. The variant has higher affinity to oxygen and shows non-cooperative behavior. I told that. So first option is correct. The variant has higher affinity for oxygen and shows cooperative behavior. It doesn't show cooperative behavior. If it has to show cooperative behavior, then it should be a sigmoidal curve. But it is a regular curve in this case. So the second option is not correct. The oxygen dissociation of the variant is less than 10% at 20 mm of Hg. That is also correct. Then at sea level, because the dissociation, that is from here, So 20 means it will be over here. It will be only 10% dissociated or less than 10%. That is why the oxygen dissociation of the variant is less than 10% at 20 mm of Hg. It will be less than 20% because of its high affinity for oxygen. Then fourth option. At sea level, approximately 150 mm of Hg, animals with this variant of hemoglobin can survive better than the ones with normal hemoglobin. Because this variant of hemoglobin, it is having higher affinity for oxygen, it will not be released very easily. So these animals will not be able to survive better with the variant hemoglobin than the normal hemoglobin. So option 4 is also not correct. So the answers are option 1 and 2. 1 and 3. Then next one. A hospital witnessed an outbreak of nosocomial infection caused by Klebsiella pneumoniae. It showed resistance to antibiotics generally used for its treatment. Under the given scenario, the antibiotic resistance of this organism can be attributed to the presence of antibiotic resistance gene acquired through horizontal transfer from other prevalent strains in the hospital. Yes, that could be one reason. Silent mutations in target sites of antibiotics. That is also not correct. Neutralization of antibiotics by enzymes that target and inactivate these antibiotics. That is also correct. Reduced permeability of these antibiotics due to the formation of a protective barrier. That is also correct. So option 1, 3 and 4 are correct. Then next one. In the ABC model, the acquisition of floral organ identity is regulated by the floral homeotic genes, namely ABC. A wild type flower is depicted below. Consider the following mutants: loss of gene C, loss of gene A, loss of gene B, loss of gene B and C. Possible floral phenotypes, that is P2S, these ones, resulting from these mutations are given below. Based on this ABC model, the correct option is or R. This is a very easy question. Everything they have given in diagrams. So you just need to check the diagram and find out the answer. But before that, you should know one thing. So if in this case, this is the ABC model, and you can see that A give rise to sepals, A and B together give rise to petals, whereas B and C, they together give rise to stamens, and C alone give rise to carpels. That is what I have written over here for easy understanding. Now if there is a loss of gene C, now C alone is lost. So naturally carpels and stamens will not be produced. You will have only sepals and petals. So these two will not be produced because C is mutated, and only sepals and petals will be produced. So such a diagram you can see in option R. So 1 corresponds to R. 4 corresponds to. Now fourth one is loss of genes B and C. Now if there is a loss of genes B as well as C, B codes for petals as well as B is involved in stamen production and C is involved in carpel production. So what will be left out? Only A alone will be expressed. So sepals alone will be produced. So that is seen in diagram Q. So this fourth one, it corresponds to Q. Then second one corresponds to S and 2 corresponds to Q. 2 is not Q. 1 corresponds to S, 1 corresponds to R. So that option is not correct. Now see the third option. 2 corresponds to P. What is 2? Loss of gene A. Now A is lost, means sepals and petals are not produced. You will have only stamens and carpels. So it will be P. Only stamens and carpels are present. Now third one corresponds to P and fourth one corresponds to R. We have already got all the four of them. So 3 corresponds to P is not correct and 4 correspond to R is also not correct. So the options here are 1 and 3 which are correct. Now next one. Auxotrophs are organisms with mutation that have lost the ability to synthesize essential organic compounds and hence need supplements for growth in minimal media. Now prototrophs are organisms that are able to synthesize essential compounds and hence can grow in minimal media without supplements. Colonies from 6 different strains of E. coli U, V, W, X, Y, Z are grown separately on a single master plate containing complete medium. That is this. This is the master plate containing with complete medium. No strain carries more than a single auxotrophic mutation. So each strain is having some kind of auxotrophic mutation and that too only single auxotrophic mutation is present. Replica plates are then made from this master plate. Then replica plating has been done in 1, 2, 3, 4. Each of this replica plate contains minimal medium to which a single supplement, either P, Q or R or S, is added. In the diagram below, filled circle represent colonies that grew on medium and absence denotes failure to grow. So in the master plate, that is complete medium, all organisms are growing. All your UV, W, X, Y, Z are growing. Now after replica plating, you can see that only some organisms are growing. But if you see all the plates, W is common. Okay, W is common. So in all the plates. So it is prototrophic strain because it grows on all the plates. Now next option 1. Okay, based on the scheme, the correct interpretation is or R. So W strain is prototrophic. So that one is correct. And strain V is auxotrophic for Q. Now where is V present? Yes, V is present over here. Minimal medium with Q supplemented. So minimal medium with Q supplemented. So V requires the medium to be supplemented with Q. So it is an auxotrophic strain. So it is auxotrophic for Q. So the answer here, first one is correct. Now strain Z is auxotrophic for R. So where does strain Z grow? When to a medium supplemented with R. So strain Z grows there. So strain Z is auxotrophic for R and strain Y is auxotrophic for S. This is Y strain and it is auxotrophic for S. It grows in minimal medium with S only. So that is also correct. So first and second options are correct. Then next question. Energy diagrams allow ecologists to compare patterns of energy flow through the trophic levels of different ecosystems, while biomass diagrams allow them to compare the amount of material present in living organisms at different trophic level. The energy flow and biomass diagrams for few ecosystems are given below. Choose the correct statements. So first option 3B, that is this one, cannot be biomass pyramid of an open ocean since biomass pyramid cannot be inverted at any level. The pyramid of biomass in ocean is generally inverted because the biomass of fishes, it far exceeds that of the phytoplanktons. So option 1 is not correct. Then next option 1B and 2B are the biomass pyramids of a grassland and a forest ecosystem respectively. Since most of the biomass in a grassland is found in the green parts and hence the efficiency of energy transfer from producer to primary consumers is higher in grassland as compared to forest ecosystem. So it is lesser in grassland compared to forest ecosystem. So option 2 is also not correct. And option 3. 3A could be, that is this one. 3A could be the energy flow pyramid for an open ocean. That is correct. Energy flow for an open ocean. Here, then option 4. 1A would be energy flow pyramid for a forest ecosystem, since the majority of the biomass is tied up in wood and is not available to most herbivores. So 1A is also correct. So option 3 and 4 are correct, whereas option 1 and 2 are not correct. So that's it. If you like this channel, please do like, share and subscribe. Thank you.